Welcome back to Dr. Kala Edu. So today we are going to discuss the topic on linkage. Okay, so what is linkage? The basic things about the linkage we are going to discuss today. So before that, a small hint that parallelism. Last video I told you about the thing that what and all the scientist has put forward or postulated the ideas, and finally Newton has postulated an idea that is called as parallelism. He told that Mendel sort of inherited behavior and this chromosomal behavior are both the same. He was telling the same. So according to that, they have listed some parallelistic characters between the Mendelian factors and the chromosome behaviors. What are these? So Mendel told that alleles occur in pair. So we used to write in the genetics, you all know that we used to write only in pairs. So TT, RR, like that we used to write in only in pairs. Same way here the chromosomes also occur in pairs. There is no difference in that. Because in Mendel's period he doesn't know that what is the things, either cytologically or nucleotically, he don't know scientifically, but he has arranged something which is exactly similar to the characters coherent with the chromosomes. So next point is we have both similar or dissimilar alleles. They are separated. How? While gametic formation are similar or dissimilar gametes, both they are separated. We all know that if it is gametic formation, T, like this. Okay, so here also during meiosis, reproduction, during the time of reproduction, meiotic division is undergone. So undergoing meiosis, what is that? It is an reduction division. Here the diploid condition is going to be converted into haploid condition. So here also the homologous pair of chromosomes are separating. So this is an diploid chromosome, and when it is got separated, it is an haploid. So both are the same. So the third thing is assortment independently. The characters are assorted, separated independently. So here also in chromosomal behavior also during meiosis there is an independence for the separation of this haploid set of chromosomes, but here they have found out one more new thing that is called as linked genes. These linked genes, they don't have assortion at all. They will not assort. They will present where it is. So they are not going to cross linkage. So what is this linkage? We are going to see today. Okay. So the linkage. What is this linkage? First, we all know that genes are determined characters. So the genes are the things which are going to determine our particular characters. And another thing is the genes will be present in the same chromosomes or in a different chromosomes according to that particular organism. So we used to know that there are so much pairs of genes in all different plants. For example, in maize we are having 20 chromosomes, and in onion we have 16. Like that we have different types of sets of chromosomes. So here not in a same place or in a genes, it may present in different chromosomes also. That's depend upon the organism. And the third point is it can assort independently. All these related to Mendelian characters, including this assortment independently. But this some not assert. This is an exemption character of the Mendelian character. According to Mendel, what he is telling, law of independent assortment. So there is an assorting, that is shuffling or separation is there. But some of the places these things are not happened. So this is an exemption. So that is some of the plants or animals we see there is no assorting, independent assortment. So what is this? So this was studied by Batson and Punnett. Okay, again. So William Batson and Punnett. Okay, they both has come across experiment using sweet pea. Sweet pea is what? Lathyrus odoratus. Scientific name is Lathyrus odoratus. So in the sweet pea, they have taken an experiment. What is this thing is homozygous purple flower with long pollen grain. What is this? Purple flowers with long pollen grain and red flower with round pollen grain. They cross both the characters. Purple flower with long pollen grain and red flower with round pollen grain. As we all know, F1 is heterozygous and obviously it will be a purple flower with long pollen grain according to Mendel's law of independent assortment. Okay, so there is no confusion till here. What they did, they crossed the F1 generation, F1 with double recessive parent. What is this double recessive parent? PL. So they crossed with double recessive parent. So if a character, when we use double recessive parent, what is it? It is an test cross. Okay, so they used test cross. What is the, according to Mendel's test cross, in F2 the ratio is 1:1 in monohybrid. When it comes for dihybrid, 1:1:1:1. But surprisingly, they come to know that there is no existence of this 1:1:1 character. So here they found that the genes are not allowed separate. How? In this F2 generation also, they got purple and long pollen grain, red and round pollen grain. Okay, so purple, I will write it fully itself. They doesn't got purple with long pollen grain or red with round pollen grain. Okay, so according to 1:1 ratio means we can get purple and round pollen grain also. Red and long pollen grain may also be the possibility if there is an existence of independent assortment in that particular character. But in F2 generation, surprisingly, they found only purple flower with long pollen grain, either red flower with round pollen grain, but they don't have a recombination of purple and round pollen grain, red flower with long pollen grain. From this thing, they have found that the genes are not allowed separately. That is, independent assortment is not taken place here. Then they came to know that what is the thing? What is the thing? Why it has happened? They have started speaking about. So they arise and thing is called as linkage. So linkage means nothing but tendency of genes stay together. So they will not getting independently assorted. So the tendency of the genes which are going stay together during the separation of chromosomes. That is very, very important. During the separation of chromosomes, while the separation taking place, is taking place, what will happen? There is no state, they will have the tendency to stay together and they will not separate each other. So this is called as linkage. So linkage means linking. So what is the thing? So the two genes have a tendency that they were placed so closely near and they will not separate is called as linkage. So next is linked genes. So they have given two names with the linkages, linked genes and unlinked genes. So what is this linked genes? So when the genes located close together, okay, so when the genes located close together on the same chromosome, you should be noted that on the same chromosome. So if the linked genes means the genes which are located close together on the same chromosome is called as linked genes. So this is from the definition linkage. So what is this unlinked genes? So they are separate apart. They are separate apart in the same chromosome is called as unlinked genes. Okay, so this condition is also called as synteny. Okay, this condition is also called as synteny, unlinked genes. So hence it is this unlinked genes also called as synthetic genes. Synthetic genes or unlinked genes which are separate apart in the same chromosome, and such condition is called as synteny. Okay, so next is kinds of linkage. So Morgan has put forward a thing that he has come to know there are two kinds of linkages here. So what are these linkages? So one is complete linkage, another is incomplete linkage. From the word itself we can understood that linkage means what? What is linkage? So the genes will be close together. So complete linkage means the genes are located very close together and there will be no separation at all. Okay, so there will be no separation at all. So hence we call it as complete linkage. So this complete linkage phenomenon is observed in male Drosophila. They come to know that in male Drosophila there is no linkage at all. Absence of linkage in some of the species of male Drosophila. This was discovered by Bridges. Okay, Bridges is a scientist. He only discovered this. Okay, next is incomplete linkage. So incomplete linkage, what is this? Is the genes will be sufficiently apart. Okay, on the same chromosome is called with, but here so that as it is apart, what is this crossing over is possible here. So the crossing over will be possible here, and this was discovered by Hutchinson in maize. Okay, this was discovered by Hutchinson in maize. So this is the two kinds of linkage. Okay, so here what is this is, because of this incomplete linkage there will be a parental and non-parental new type of combinations can be arised as the genes are located far apart. And one more thing is how they find the link, it is an complete linkage or incomplete linkage, or either it is a linked gene or unlinked genes, means if there is a chance of 50% recombination. So there is a calculation is there. Okay, so if they find the recombination is more than 50%, then it is said to be unlinked genes. So if the recombinance percentage is below 50%, we come to know it is unlinked genes. The genes are held to close together. So more than 50%, less than 50%. If it is towards right side, it is unlinked genes, and towards its less, it is called linked genes. So one more thing here is that is linked groups. So linkage groups we used to tell as linkage groups. That linkage groups are nothing but these linked genes are linearly, they will be linearly arranged in the particular chromosome, which is called as an linkage groups. And linkage groups are very specific, that the number of linkage groups and number of the chromosome which is present in the haploid sets are equal. Okay, you should know that the number of linkage groups and the number of chromosome in the particular haploid set are the same number. Okay, in this video I think you will be clear about the linkage and what is linkage. If you have any doubts, you can comment me in the comment section. I will clear your doubts. We'll meet you in the next video. Thank you.